Okay, let's get started. Hi, everyone. Welcome to this Unisense Microsensor Workshop. My name is Line Daugaard, and I am Product Manager here at Unisense. It's really great to see that so many have joined us for this workshop from many different locations all over the world. And we are looking very much forward to have a great week with you here online. Although there'll be a lot of communication from us to you through the lectures, we also hope to interact a lot with you. So please just ask your questions along the way. The goal of this workshop is to give you the tools to apply microsensors correctly and to give you tools so that you can achieve great results with the microsensors. I hope that you will enjoy the workshop. So welcome to all of you. Yeah. Okay. So during this welcome lecture, I will just briefly introduce you to the Unisense microsensors and talk about why you should use microsensors for your research. Then I will show you some examples of different applications with microsensors. And then I will tell you a little bit about Unisense. And at the end, I will go through the workshop program. If you've had any questions, you're of course welcome to ask them, and you can also put them in the chat here. So the Unisense microsensors are small electrochemical sensors. They are handmade here at Unisense, and they are made of glass that is drawn very thinly to make these small tip sizes. We make tip sizes between 500 micrometer and down to less than 10 micrometer. We can measure the analytes shown up here. We have amperemetric sensors for oxygen, hydrogen, nitrous oxide, nitric oxide, and hydrogen sulfide. And then we have potentiometric sensors. These are the PAs, the redox, and the electric potential sensors. We also have temperature sensors, and we also sell optodes for oxygen measurements. The signal from the microsensor is very, very small, and therefore you need a high quality amplifier for amplifying such a tiny signal. We produce all the electronics and software for the microsensor measurements in-house here at Unisense. So why should you use microsensors? First of all, you will get real-time measurements. This means you can follow the signal from the sensor all the time. And because of the small tip size, you get a non-destructive and a minimally invasive measurement. You do not disturb the natural chemical gradients in the sample, and you can obtain a high spatial resolution. And the rule of thumb is that the spatial resolution equals the tip size. You also get a very fast response time, and the microsensors, they have a low analyte consumption and thereby also a low sensitivity to stirring. Our microsensors are used in many, many different applications, from environmental research to biomedical research, and also in chemical applications and much, much more. So I will now show you some examples of different applications with microsensors. In biogeochemistry, sediments and biofilms are extensively studied with microsensors, both in the lab and also in the field. The microsensors are used to obtain high-resolution microprofiles throughout the samples to evaluate the dynamics of the analytes in the different layers. We sell full systems for field and underwater measurements. The microsensors can be pressure compensated so that they can be used with landers on the deepest seafloor, and we can go down to 6,000 meters. We have a mini profiler system for making microprofiles. We have a chamber lander for incubation measurements. And then we also sell an eddy covariance system for flux measurements. Tay will tell you more about these field systems in his lecture tomorrow. Microsensors, they are also used a lot in plant science to look at the root-soil interface and to look at the conditions inside of the plant tissue. And this very often requires small tip sizes. Here is an example for oxygen measurements going into the shoot meristem with a very small 3 micrometer oxygen sensor making a microprofile throughout the tissue. The researchers here, they found a low oxygen level inside of a tissue area of only 30 cells. And the researchers, they found that the production of new leaves is depending on these hypoxic conditions. Microsensors are also widely used for monitoring coral reef ecosystems, both in situ and in the lab. And here is a study when microsensors were used for monitoring coral reef ecosystems. And they were used to characterize the oxygen concentration in the coral diffusive boundary layer surrounding the coral throughout the diel cycle. In microbiology, microsensors are also used, and they are used to study both bacterial suspensions and biofilms. Here, microsensors are used to study the microenvironment inside a colony biofilm. The study shows here the oxygen profile and also the redox profiles in colony biofilm that was only a few hundred micrometers thick. Real-time production and consumption rates of microbial suspension, they can be studied in closed chambers, as shown here, with the microsensors. And this study is showing real-time hydrogen production from a cyanobacterial mutant. Our microsensors are also used in biomedicine for investigating animal models and for measuring local conditions at the cell level, and for measuring oxygen consumption rates in cell cultures. Cells growing in 3D called spheroids or organoids are also getting more and more attention because they mimic the in vivo conditions. And here is an example of measuring the oxygen concentration all the way through a 3D spheroid to characterize the oxygen availability for the cells inside of the spheroid. In neuroscience, microsensors with a tip size down to 3 microns have been used to study the oxygen tension in the brain tissue, both ex vivo in brain slices and also in living animals. And here is shown the oxygen partial pressure levels in brain tissue from a living mouse at different conditions. In cancer research, the sensors are used to measure mostly pH and oxygen in tumors. Here's a study looking at the oxygen concentration in tumors after different treatments with nanoparticles. The microsensors are also used to answer questions about animal physiology. Here you can see oxygen measurements in a fish eye, and over here you can see measurements in the large intestine of a spider. Hydrogen is getting a lot of attention these days as a renewable energy carrier, and we see many studies on light-driven water splitting using photocatalysts. And with the hydrogen microsensor, you can measure the real-time hydrogen production directly in the reaction vessel. Lastly, I will show you here an example of a hydrogen sensor sitting in a biogas reactor measuring hydrogen inside the reactor. We can optimize both the design and the performance of the microsensors to fit the needs for your application. Here you can see different configurations, sensors mounted in different types of needles, in different steel tubes, and also in different types of flow cells. We can also optimize things like the response time, the stirring sensitivity, and the concentration range. So we can really optimize the sensor to fit with your application. So now I will tell you a little bit about Unisense. Unisense was established in 1998 by researchers from Aarhus University here in Denmark. This year, we are celebrating our 25 years anniversary, so we have many years of experience with microsensors. The company is largely owned by scientists, and we are now 28 employees, and most of us have a PhD. We are also an ISO certified company, and you can, of course, find more information about us and the microsensors on our website, unisense.com. Unisense sell and collaborate worldwide. We have a network of more than 5,000 customers all over the world. We have a distributor network. We have distributors in China, Japan, Korea, India, and Brazil. Every year, we typically have 2 workshops here in Denmark. We also do an annual workshop in Asia, and we also do field systems training. And then we have these online workshops as well. We answer more than 5,000 emails every year with questions about microsensors. And if you look in the literature, you can find more than 2,800 peer-reviewed publications using Unisense microsensors. These are pictures from a recent workshop that were held here at Unisense at our new facilities here in Aarhus. And like this online workshop, they are a mixture of lectures with microsensor theory and applications. It is demonstrations in the lab and also lectures by experienced microsensor users. We always encourage people to bring their own samples to these workshops so that you can really try out the sensor with your application. We expect to host the next in-person workshop here at Unisense in Aarhus this fall. The exact dates will be announced later this spring, so keep an eye on that. This is a snapshot from a workshop. It's just to show you how many different type of samples that we see. We had researchers working with photocatalytic hydrogen production. We have researchers working with photosynthesis of corals, biogas upgrading, respiration rates of oocytes and embryos, nitric oxide and nitrous oxide production in ammonium oxidizers, biofilm conversion of captured CO2, making pH and hydrogen profiles in the biofilms. We had someone working with carbon sequestration in coastal ecosystems. We had respiration rates of foraminifera, nitrous oxide emissions from wastewater, oxygen measurements in plant tissue, oxygen levels in vivo after neurostimulations, and oxygen and sulfide at the sediment water interface in situ. So many different applications.